Hello students, now we will see about Morgan's second experiment that was done by selecting another two characteristic features, that is color of the eye and also size of the wings. In previous part 17 video, I explained you about Morgan's first experiment that was done in 1910 by selecting two characters, that is body color and also eye color. Now we will see about the second type of cross. So cross 2. That was done in 1911 year. T.H. Morgan selected two characters in Drosophila. What are those two characters? One is color of eye. Second one or second character is size of wings. So under this color of eye, we know that the dominant feature is expressing red eye and the recessive feature is expressing white color eye. I told you red color of the eye is the natural color of the eye that is existed in the nature. Coming to size of the wings, the natural one is indicated with a dominant allele like this form, and it is nothing but large sized wings. And recessive one is miniatured wings. As I told you initially, in nature there are red color eyed and large sized wings, Drosophila, existed naturally. Now let's take initially the parents. With what type of genotype are existed? These are of X chromosome of the female. When genes are giving out so and so character, there will be dominant alleles and also recessive alleles. That means gene is getting expressed in contrasting forms. When it is crossed with normal red color eyed and large sized wing male, here it means that this is X chromosome of female, this is X chromosome of female. Here this is X chromosome of male, this is Y chromosome of male. How can we write this one? When dominant alleles and recessive alleles are present in the same type of chromosomes or in the organism, then it is considered as heterozygous female. Now here there is only one X chromosome, so we can call it as hemizygous male. If you extract the gametes, it will be giving this is one gamete, another gamete of the X chromosome. Next, X chromosome of female, and this is Y chromosome. Now let's write F1 generation. Now the offsprings obtained are, if we cross this X of female with the X of male chromosome, then there will be homozygous female. If the cross is like this, then it is homozygous female. Under cross, the dominant alleles with Y chromosome, this is considered as hemizygous male. This is showing parental combinations, or we can call it as this type of organism is showing parental combination. This is also parental combination, because we are seeing red color eyed and large sized wings type of phenotypes in the offsprings. The next one, red color eyed, large sized wings, again heterozygous female. heterozygous female. This is also showing parental combination. The last one, if we write, it will be the recessive type of alleles are binded with the Y chromosome. Then X chromosome of female, it is fused with Y chromosome. Now we are getting hemizygous only, but it will be male. When we intercross the offsprings, here heterozygous female and also hemizygous male, that is white-eyed and miniatured wing male. Then how the gametes are going to be? So we will intercross heterozygous female with white-eyed plus miniatured wings male. So the allele distribution is going to be from, this is female organism, this is X chromosome and this is also X chromosome. We are crossing with white-eyed male offspring from the F1 generation. We are taking this and also we are taking this offspring and we are intercrossing. So how gametes are going to be here? This female organism can also be written as, the allele distribution can also be written as, this is indicating the dominant alleles and this is recessive. When we are crossing any type of organism with purely recessive type of alleles, then we call such type of cross as test cross. It means in the F2 generation, we are considering F2 cross. So here the gametes can be expected as dominant allele with one dominant allele. So one gamete can be having both dominant alleles, another one. Dominant allele plus recessive one, another gamete. Recessive allele with a dominant one, another type of combination of alleles, that is recessive one with a recessive miniature wing allele. Now, the gametes from male can be of only one type, that is a recessive one and also Y chromosome. This will be the allele distribution. Now we have got from heterozygous female 4 different types of gametes. And here obviously it will be only 2 types of alleles or gametes. The gametes like this, one type, that is another type, that is third type, and fourth one purely recessive, which is crossed with one type of gamete from the male and a Y chromosome from male. Now we are crossing these 4 with these 2 types. So how the offsprings are going to be in F2 generation? When we cross these both, X chromosome of female, X chromosome of male. This is considered as heterozygous female. Next, let's cross this one with Y chromosome. So 2 dominant alleles of X chromosome cross with male. Now, let's do the another type of cross, that is one dominant allele, one recessive allele with that of Y chromosome. So we have got a special type of recombinant here. Now, let's cross this one with a purely recessive form of alleles. So it can be like this. Here also we have got one more recombinant. Now, let's cross this both. M plus, this is X chromosome of female crossed with X chromosome of male. This is also considered as the recombinant because there is only one dominant allele in the X chromosome. Let's see another type of cross with a purely recessive one of the X chromosome of a female. X chromosome of female when it is crossed with X chromosome of male. Next. Again, this purely recessive one is crossed with Y chromosome. When we observe the F2 generation, the parental combinations are not only simply 4 here and 4 here. It means that for our convenience, we have written about a few number of parental combinations and few number of recombinants. Here the percentage is 62.8% of parental combination, and coming to recombinants, the obtained percentage is 37.2%. So the parental combination is okay, more comparing to the recombinants, it is more. But when we compare the first cross, that is body color and eye color, the percentage of recombinants has been increased. There, the recombinant percentage is 1.3, but here it has been increased up to 37.2. By this experiment, we can understand that there is less linkage between the chromosomes. Means there is distance, more distance between the alleles that are present on same chromosome. It also means that linkage group is less in case of such type of characteristic featured alleles. So that we have increased the recombinant percentage comparing to the first cross. The same type of cross and same type of offsprings can also be obtained when we cross between purely recessive form of parents. These are actually obtained in the F1 generation. Also, their cross is like this. These offsprings we can see in the F1 generation also. According to NCERT textbook, page number 84, they have given the cross like this. Still we will get same type of combinations. So you please go through the combinations. For your understanding, I will write the cross again. If we consider this one as parents, according to NCERT given type of cross, still we get the gametes as one type from the X chromosome of a female, another type, and another type X chromosome of male, and also Y chromosome. In F1 generation, we will get the offsprings, one type, another type, third type, and fourth type. But here, from the F1 generation, the selected offsprings are heterozygous female and also purely recessive one. This one is selected and this one is selected from the F1 generation. Still the cross is same. Still we get the allele combinations as cross with this type. And gametes obtained can be the same which I have just written before. Third type, fourth type. Now cross with purely recessive type and also X chromosome. Still we get the parental combinations. It is, I already said, it is not like just 8 number parental and 8 number. The percentage is different because it is not possible for the fruit fly or Drosophila to consider only 4 number and all that. There will be many number of flies. Parental combinations percentage is 62.8 and also recombinant percentage 37.2%. So this is about the Morgan's second type of cross. Next topic is gene mapping. Gene mapping, also called as linkage mapping. It is also called as chromosomal mapping. No. Gene mapping is given by Alfred H. Stewart Went. How can we define the gene mapping? Gene mapping is defined as establishment or identification of linked genes on a chromosome so that we can know the percentage of crossing over. As the establishment or identification of linked genes on a chromosome so that we can know the percentage or the frequency of crossing over. What does it mean? It means that we already learned that linked genes in the sense, if one type of allele is present on the chromosome, another type of allele is present, then we can say the distance between the linked genes here according to here, it is more, so that there is more chance of crossing over with the help of homologous pair of chromosomes. Now, if I write another pair of homologous chromosomes, here if you see the distance between the alleles on the chromosome or on the loci of chromosome, then it is very less, so there is very less chance of crossing over with the help of homologous pair. We can also represent here crossing over between linked and non-linked genes in another diagrammatic representation also. If we take one pair of homologous chromosomes like this and another type of loci, so this is one loci, this is another loci. And what type of allele distribution we can write here? Purely dominant one will write, purely recessive one here, dominant one here. On the means these are present on different loci. Since here there is more distance between the alleles, we can write like this arm or chromatid can have cross with this homologous pair of chromosome, and there is more chance of crossing over, so that we can obtain more number of recombinates. Recombination frequency can be defined as the number of recombinant offsprings obtained in a cross. So we can define recombination frequency like this. In case of recombination frequency, the crossing over results in formation of many number of variants. It can be of many number also. That number we are including under the term recombination frequency. Next, so we know that how the recombination happens. Recombination happens by breaking up of DNA and recombining with new DNA fragments. Next, this recombination frequency is explained by one type of model, that is Holliday's hybrid DNA model. But in detail explanation is not necessary here. Just the term is enough for your NEET point of view. Now, there is a formula to calculate this recombination frequency. So, recombination frequency can be formulated as number of recombinants divided by number of total offsprings into 100 to extract the percentage, recombination frequency percentage. Mapping significance. Gene mapping significance is to understand percentage of crossing over. Now, and also genetic complexity. Here, percentage of crossing over is depending on distance between the alleles or the genes on a chromosome. It depends upon distance between genes. We can also say linked genes on a chromosome.